So let's see after the chaos if, yes, that looks good. Thank you very much for coming. It's a pleasure to be here today and to talk about an area of marine science that actually lacks attention, but that is important and that might actually become even more important as our oceans face pressures, multiple pressures due to global change. And the white elephant in the room are jellies. And being at the Jellyfish Bloom Symposium and in India, I know that you're aware about this. So thank you very much for organizing this meeting here and for inviting me. But I think we still have a far way to go to actually convince not only the public, but the scientific community that jellies are important and deserve more research attention. Throughout today's talk, I would like to visit some paradigms about gelatinous zooplankton blooms and look into the population development of native, but most importantly, of invasive, non-indigenous jellies, because they are the true troublemakers. Then I would like to remind you about some key traits jellies display and that actually might allow them to proliferate under global change. Then it's very important to actually consider that climate change has some major impacts on marine ecosystems, and I would like to pick one positive example, how this can also lead to a higher production and carbon export in the future ocean, before I actually close with some perspectives and innovative approaches how to actually address jellyfish research and jellyfish blooms. So jellyfish, or jellies, are primarily lumped into two big groups, which is the true cnidarian jellies that have nematocysts, but we also have the comp jellies. And people knowing me, they know that these are my guinea pigs, and there are some others in the room that have the same. But if we want to understand jellyfish blooms, we actually also need to consider, this sound is very bad or what? Is it okay? Okay. But if we really want to understand jellyfish blooms, we also need to consider other bloom-forming gelatinous zooplankton organisms, and these are pelagic tunicates. And pelagic tunicates are very important because they can form dense blooms. So I request you as a jellyfish community to broaden your perspective and actually also include these members, because they could actually be the ones that are profited under global change. This is a very common picture when people talk about jellyfish. This is kind of what they will come up with. It's, bloom forming and human impact interactions. But it's important to actually remember that it's part of the natural life cycle of jellyfish to bloom, to actually form spawning aggregations and to maintain genetic diversity. So that's why when we see these images, it's not purely human impact that is kind of leading to these jellyfish blooms. It's part of their natural life cycle. Irrespectively, it has been raised by a lot of prominent journals and newspapers that we are living in an era and facing an increase of jellyfish populations, which lead to increasing human impacts. But the thing is that these paradigms, and also the dead end paradigm, like Kylie really nicely illustrated yesterday with her parasite study, is kind of coming to an end. And the thing is that we basically have a very scarce knowledge about jellyfish population dynamics because they're disregarded in most food web studies. Long-term data series are very, very rare. And the top-down control, so what is controlling the populations, is basically unknown. So diving into the first aspect, if you look at this global map by Lucas Brotz, you can see that it's depicting in different color code the trend of long-term jellyfish population. And in dark red, you see time series with a high likelihood that jellies or jelly trends are increasing. However, there are a lot of areas where there is a low certainty of jellyfish increase. There are areas where there is basically no change or even a decrease in the jellyfish abundances. And most importantly, if you zoom into one of the high likelihood jelly increasing time series in the Bering Sea, you can see that over the past 30 to 40 years, there is huge cyclicity in the data set. And depending on where you are snapshotting this time series, you can see a dramatic increase, a dramatic decrease, or basically no change. So this highlights that on a global scale, we really need to understand what are the mechanisms behind these fluctuations. But what we know for sure is that the non-indigenous species are a real matter of concern. And this is an image, a drone image. You'll see a video in a short sequence. It's Rhopilema nomadica. It's listed as one of the most troublesome non-indigenous species by the European Commission. It invaded the Mediterranean Sea in the 1970s, and it's now forming dense blooms every year. And this drone video illustrates you the extent and the magnitude of these blooms that they are observing in Israeli waters every summer. So here you can really see that jellies are causing a very problematic, local impact. But this is not the only one. We have Blackfordia virginica. We recorded it lately in the southwestern Baltic Sea. It's a brackish water species that actually likes low saline waters, but it's distributed around the globe, and I'm pretty sure it's very widespread, but people simply didn't look for it so far. There are other species, Anopilema, but also Miniopsis leidi, and this is a small sequence from the southwestern Baltic Sea, where you see different life stages of Miniopsis occurring together, adults, lobates, but also tiny larvae. So all the dots that you see in the background, they are larvae roughly 1 to 2 millimeter in sizes. And we can reach population densities of up to 1 individual per liter, which means 1 animal per milk bottle. If you look at this map, this is the range expansion of Miniopsis across Western Eurasia. Actually, most people contributing to this study are sitting in the room. It's a collaborative effort with more than 12,000 data points on presence-absence of Miniopsis throughout Western Eurasia. But if you zoom into the northern European area, it's striking that you have hotspots along the European coast where you have very high abundances that are kind of in close vicinity to areas where Miniopsis has not been present. And here, this is to highlight as well that recording the absence data is very, very important in order to understand the mechanisms, what's actually going on here. So we teamed up with physical oceanographers and did ocean current modeling. So in white, you see depicted the ocean current and the velocity. But we thought we also need to know what is actually the stability of these ocean currents. So with the stability here indicated by a color code, warm red means it's like a hose that you turn on, the water's going in one direction, whereas when you go to purple, it's like a kid sitting in a bathtub and playing, so the water's splashing back and forth without a net transport. And when you look at this, you see that there is a southward current along the UK coast, which actually drifts the animals back to the European coast, even if they might arrive there once in a while. Whereas there is a very persistent current along the continental European coast. So we wanted to know what is actually the extent and the importance of ocean currents for seeding planktonic organisms. And for this, we used Miniopsis as a model species because we were fortunate to have a very good database and data set at hand. So in the period from 2007 to 2010, Miniopsis was widely distributed, here indicated by red points. But then we faced a period of 3 very, very cold winters. And during this period, Miniopsis disappeared from most areas, not only of the central Baltic, but also Sweden, Norway, and was retracting to the warm areas in the North Sea. However, one warm winter in 2014 was enough that Miniopsis recolonized the entire area. We then used microsatellite data to actually confirm if this was a unique invasion or if we just missed animals during, the cold period. And if you look at the genetic structure before and after the cold sweep, you can see that it's a new genotype that arrived. And this actually indicates that that is a unique, new invasion event. And more strikingly as well, during 2014, when we analyzed samples throughout the entire distribution range, covering Belgium all the way to Norway and the central Baltic, so several thousands of kilometers, there was no population structure at all, which indicates that ocean currents really drive very, very swift recolonizations. And if you want to halt non-indigenous species transport in the long run, it's these highly interconnected areas along the European coast where we have very, very high port densities that actually need to be managed. But looking at this map again, it becomes also apparent that there is a large area here in the Baltic where Miniopsis is absent. This is a very low saline area, below 6 PSU, down to freshwater. But Miniopsis is present in the southern range in regarded to be low saline areas, even though the salinity of the Caspian Sea and the Black Sea is higher than what we observe in the Baltic Sea. However, we conducted different field experiments in situ reproduction rates, but lately this is the final proof of evidence that salinity has a high impact on reproduction rates. So what we did is we incubated animals of the same size, acclimatized them over 2 weeks at certain salinities, fed them constant, similar food conditions, and then we checked the reproduction rates under the different salinity levels. And as you can see, at the low salinity level that's characteristic for the Baltic, there is basically no active recruitment. So the lack of Miniopsis in the Baltic can be associated to the fact that they basically can't reproduce. So they must spend their energy on osmoregulation instead of reproduction. Where does Miniopsis come from? So it's native to the east coast of the US, actually entire Americas. But the population that's present in the Gulf of Mexico and further north in New England, they're the ones that were actually seeding the populations in Western Eurasia. So the one from the Gulf of Mexico seeded the Black Sea and the Mediterranean Sea, and the one from the northeast coast came to Europe, to northern Europe. This is a dendrogram based on 72 whole genome resequenced animals throughout the distribution range. And you can see that there is the population structure is very similar in the northern range, though significant, but there is a large population differentiation in the southern range. And this is true for both the original invasion into the Black Sea and also the later invasion to the Mediterranean Sea. The nice thing about whole genome data is that you can reconstruct the past. And we did that by so-called PSMC analysis. So basically, each individual that we sequence will in a second be displayed on this map. The color of the animal will depict its origin, so either in the native or in the invasive range. And then you see how the population, how its population density changed throughout time. You have past on the right-hand side and present on the left-hand side. And if you look now at this very confusing plot, you see that back in time, all animals show ancestry. But then interestingly, there was a period of very rapid effective population growth, which is kind of corresponding to the expansion in the native range along the US East Coast. And now when you zoom into the more recent history, where these lines, the red line and the green and yellow diverge, that actually indicates the invasion into the Black Sea. You also see that all animals that are present in the Black Sea originate from the southern population, and you see that the southern population showed a dramatic increase in the effective population size, meaning that they actually grew and expanded. In contrast, when you look where the green and the yellow line diverge, so the secondary range expansion to the Mediterranean Sea, this is purely from Black Sea animals. And you see a dramatic decrease in the effective population size, which is indicative of a population bottleneck. For the north, however, both populations follow each other very closely. So if you take together these results, one can conclude that there are small but recurrent introductions from the native range in the northern part to northern Europe, whereas for the south that was a large but time limit introduction, whereas the secondary range expansion to the Mediterranean Sea was a very, very severe, represent a very severe bottleneck and was due to very few founding individuals only. So if you look at this video now, which is from the beach where I'm living, we see Miniopsis blooms every summer. And all these small dots are again small larvae, and you see larger animals in the background. So I really think it's important that we combine different approaches in order to understand what's happening out in nature and what we observe. But coming back to the second point, what about jelly traits? And it's a pity that Mike is not here because he was very interested in traits. But I would like to remind all of you that jellies and gelatinous zooplankton in general have some really cool characteristics that make them actually likely to thrive under global change. And one of this is the podocysts. So basically, polyps under unfavorable conditions can form dormant stages and survive very, very tough conditions on Oxia. And then under good conditions, they just wake up again and continue their business as usual. And what has been described by Haeckel in 1881 already, they can also shortcut the life cycle. So basically, polyps can settle for a few days only and then metamorphose directly into an ephora, leading to a secondary peak in the population. And this has lately also in 2019 confirmed from Japanese waters, and I think it's very important that we also consider this very, very old knowledge that has been around for a long time in order to understand and explain the observed pattern that we see nowadays. Life stage reversal, so basically some jellies can also go back in time, so they can become from an adult to a juvenile again. And a very prominent example of this is Turritopsis. And here you see on the small table, that's a 2-year period where a Japanese colleague managed to re-juvenilize one individual 10 times. Isn't this amazing? Basically, that's the forever growing jellyfish. But also ctenophores are cool because they also have mechanisms by which they can actually shortcut the life cycle. And this has been known for a very long time that larvae can reproduce. So instead of going through metamorphosis, they can stay as a larvae and reproduce. And Juan actually had a very prominent culture in SARS in Norway, only keeping the larvae because that was much less time consuming and easier to get eggs from instead of letting the animals grow to adults. And going back to literature, it was actually Chang in 1882 or 92 that suggested that larval reproduction is a key trait of ctenophores that allows them to survive unfavorable conditions. But this hypothesis has been untested for a lot of years, actually more than 100 years, because in 2010, I think it was, we were the first to confirm that Mertensia in the Baltic Sea is exclusively reproducing in the larval stage. So reproducing few eggs right after birth keeps the population going and is more beneficial for them than growing up. And we suggested that this might be due to predation pressure, because if you do the life history model, you can see that a population can only be maintained if you mature as early as possible if the predation pressure is too high. Regeneration is a big topic, and here some ctenophores can regenerate from few cells. But we also have Miniopsis that can regenerate not only body parts, but half the animals. They can regenerate from a quarter of an animal. They can stay as stable half animals. So very, very intriguing results. Mark Martindale has been working on that. But we also found that Miniopsis shrinks. They not only shrink, but they shrink to keep up egg production. And this is pretty cool because when they shrink and channel body carbon to the next generation, this next generation has a totally different food size spectra. So that way around, they can get over periods that are unfavorable. And this shrinkage is actually a fact that all or nearly all jellies can do, so it's really something to consider and makes them very unique. And then last but not least, we have the balloon effect. And this is basically that we have huge bodies and very low carbon. On average, jellies can be approximated as having 0.5% carbon of their wet weight. And here I talk about wet weight because that's not impacted by salts. But if you go to low-weight ctenophores, it's only 0.05%, so basically there's nearly no carbon. However, if you look at this map from José Acuña and Jack Costello, you see the body carbon on the x-axis, and then you see the feeding standardized for the carbon content. So basically, you get rid of the water in the jellies, and then you compare the feeding just on base of the carbon content. And if you do that, you see that jellies have exactly the same feeding rates. They're lying on the same slope like fish. But fish are very young and highly evolved. But jellies, being very old and very dumb in their feeding mode, if you want to say that, they still have the same feeding efficiency. So I really think it's important that we open our eyes that there are these mechanistic interactions that are important. So these were just some key traits that I picked that I hope make you understand and incorporate into future research initiatives that jellies do have certain special characters that might allow them to thrive in the future. But what does the future do with our oceans? This is a niche model which is depicting the increase of the smallest algae, picophytoplankton. And you see the scale is different. You see that there is there's a factor 10 difference. And you see that throughout our oceans we expect that the primary producers get smaller. What does this mean? If you increase the microzooplankton, and we heard a very nice talk about microbial diversity yesterday, you increase actually the importance of the microbial loop in the future. And this has huge impacts on the transfer efficiency to higher trophic levels such as fish and zooplankton. But, and this is the good news, there is a shortcut in the food web, and these are pelagic tunicates because they're the baleen whales of the plankton. And I picked this example because larvaceans, it's a group of pelagic tunicates, they're the most extreme ones, so they can feed on viruses and bacteria. If you throw a net in the water, it's mostly only 10% of the plankton counts that are represented by larvaceans. But if you look at their growth rate, so the carbon-specific and temperature-corrected growth rates, you see that they have an order of magnitude higher growth compared to copepods. And we have actually shown across a gradient in the Indian Ocean that these guys, the jellies, so to say, can contribute up to 5 times more to secondary production, meaning being 5 times more important as fish food compared to copepods. Still, the community believes that copepods rock the world. But I think we really need to modify this paradigm too. But larvaceans are not only important in carbon cycling and are expected to become more important as the primary producers get smaller. It's also that they're very important in carbon export because they produce houses around them that allow them to feed on the small stuff, and they jump out of these houses up to 40 times per day. And Shin also worked very nicely in his old days with some larvacean species, fantastic work. But still, they're a kind of a forgotten group. But larvaceans are not the only ones that are important. We also have other jelly groups that have pretty high growth rates. And you see that here in color code different groups of jellies and their carbon-specific growth. And I would like to pick one recent example from the Northeast Pacific, so off Alaska, where actually they found a bloom of pyrosomes in 2017. This is a tropical species that you suddenly find in the kind of lower Arctic areas, subarctic. But also some brand new results from our lab, where we have actually shown that Miniopsis in the central Baltic Sea had quite a very stable abundance throughout the past 15 years. These are November data and a standardized jellyfish count on the 45 station grid. But what we saw in this year is that the abundances peaked. And basically we have an order of magnitude higher densities this year compared to the average over the past 15 years. And we can find that for the last 4 years, which were characterized by very low, very high winter temperatures, that there is an exponential increase, actually with an exponent of nearly 4. What's the cause of this? We still don't know, because there are multiple pressures. But I think what's very important is that we are aware about these multiple pressures. We see time series, we see the fluctuation, but we really need to get going with understanding what are actually the mechanisms behind these changes. What are the mechanistic interactions? And I would quickly like to show one example from Limfjord. It's a fjord system in northern Denmark and northern Europe. And here you see the decrease in the demersal fish catches from monitoring trawling surveys. And where the fish kind of, in the period when the fish disappeared, you see a higher incidences of the trawls that couldn't be completed because they were overfilled with jellies. So in years up to 60% of the trawls that had to be kind of, discarded. So this indicates that fishing is probably an important factor driving this increase in this localized area, but there are other pressures that are acting simultaneously, and that's warming, eutrophication, toxic algae blooms. So this is just to highlight that it's very, very complex to actually understand what's happening, and this is also very local. So we really need to get going to get a global picture in place. So as a perspective and innovation, I hope that I made you convinced that we really need to start to combine approaches to get experimental evidence and to include multiple pressures. And it's very difficult, I know, to get a mechanistic understanding, but this is needed if we really want to be taken serious by the community and if we want to address if jellies are increasing in the future or not. To achieve this, I think a way forward is new technologies. And this is just the new toy. It's an optical, it's a new ICES that Ross Hopcroft just manufactured, and we can play with that in Alaska next year. Dougal and Alan are joining. This is a system, video system, that actually images 240 liters of water per second. So you can imagine that with these advances, we're suddenly able to actually depict very rare species that were otherwise not detectable with visual observation technologies. But we also need to move over to drones, for example, or satellite images. And I think that genomics and physics also should be incorporated and linked, and I think that Alan and Rebecca, there are some people out here that are doing some great work with that already. Time series are needed, everyone knows that. It's very difficult to keep them going. So that's why it's important, I think, to link up with fisheries institutes. And we need to have a change in our mindset, not only on national but on international level, to actually bring jellies on the agenda. And with this, I would like to thank my collaboration partners and mentors, and I would actually like to close with a video, and I'm happy to take questions while the video is running, of a new species that we actually found in our water. So this is just off the beach where I'm living, and 6 years ago we realized that bluefin tuna are back. So basically this is now a video that you'll see where you see the Helsingborg castle in the background and you see jumping bluefin tuna. And these tuna I picked up because they have been documented that even though it's a fast swimming and high energy demanding fish, they do feed on ctenophos, for example. So this dead end paradigm, it's really not true, and I think with this video you come to a realization that, wow, even these species where we never thought that they're jellyfish eaters, they're also among the ones that opportunistically prey on jellies. So thank you very much.